Today we will see DNA replication in prokaryotes. In my previous video, I tried to discuss what we mean by DNA replication, how this DNA replication carried out, what are the requirements. If you remember, we tried to define replication. It is the process of copying of DNA with very high fidelity. You know DNA is a double strand. So during DNA replication, what will happen is that double strand will be separated. After that, upon that, each new strands will be synthesized. So finally, two daughters DNA strand will be formed. That is what we call DNA replication. So we said the requirement is like DNA template, substrates, DNA polymerase and the primers are very important, very important. So today we'll try to explain the process of DNA replication in prokaryotes, how DNA replication is carried out in these kinds of organisms. What the role of different enzymes? What are that enzymes? How that enzymes support this process? First, let us see what we mean by prokaryotes. What are that organisms? You know, prokaryotes are organisms like bacteria, single-cell organisms like bacteria. They have no defined nucleus. Not bounded by nuclear membrane. So there is no defined nucleus. Rather, it is nucleoid, no nucleolus. Not only that, there is no other organelles like mitochondria, reticulum endoplasmic, Golgi apparatus, vacuoles and lysosomes are not found here. The kinds of chromosome found here is a circular. So DNA is a circular here. It is not a linear like eukaryotes. Not only that, there is extra chromosomal DNA. That is what you call plasmids. Extra chromosomal plasmid found here. This is what makes this kind of cell unique. So it contains circular types of DNA. Anyway, we will come again the contribution of the importance of having these circular kinds of DNA in managing, in controlling DNA replication, in termination of DNA replication. We will see anyway. So anyway, organisms having these properties, what we call prokaryotes. Organism having such types of cell is what we call a prokaryotes. So DNA replication takes place freely in the cytosol because there is no nucleus in the cytosol, freely in the cytosol. There are different enzymes. The first three polymerase enzymes are DNA polymerase 1, DNA polymerase 2, and the DNA polymerase 3. The main role of DNA polymerase 1 is it is very important to remove a primer. Last time in my video I tried to define a primer. It is a short stretch or short strand of RNA that is important to start replication. After that replication is begun or started, it will be removed immediately. So that removing it is the role of DNA polymerase 1. So this DNA polymerase 1 remove primers after DNA replication begin. DNA polymerase 2 take part in repairing. After primers removed, it involved in repairing that site. DNA polymerase 3 is required for DNA synthesis. It adds new nucleotide on growing strand, growing strand at the 3' end. Just new phosphate or new nucleotide will come and attach at the 3 hydroxyl group of sugar, then again phosphate will come and attach at that region. In such a way, elongation can be carried out. So in that case, DNA polymerase III involved. The question is, how does DNA replication begin? Replication begins at the origin of replication, or we call it origin of replication. How we can detect that origin of replication? How we can identify? You know, approximately there is 245 base per long end. It is rich in 80. It is rich in 80. It is rich in 80. Adenine and thymine. A sequence of adenine and thymine like that. So that region is what we call origin of replication. What are the mechanisms? There are different mechanisms, generally three mechanisms, three processes. The first is initiation, elongation, and termination. Let us see in details of each process. In initiation, there is a certain proteins that recognize and bind to the origin of replication or ORIC. It recognizes that region, the region having 80, the region which is rich in 80 nitrogenous bases. So it detects that region and bind to that region. After that, the complex will be formed. So replication bubble is formed. Something bubble is formed. Then DNA winding begin. So helicase enzyme cut the bond, open up the DNA that are extended by the directional, forming what we call DNA replication fork or Y-shaped structure. Just helicase cut like a scissors. It opened the strand. So finally, Y-shaped, the fork-like structure will be formed. Not only helicase, there is enzyme we call single-stranded binding protein. This single-stranded binding protein, it coats the DNA around the replication fork to prevent rewinding of DNA. It coats that single-strand DNA, so there is no chance of rewinding. It coats each strand, so there is no rewinding of DNA. There is also topoisomerase. This topoisomerase bind at the region head of the replication fork to prevent supercoiling. It makes loose. It does so by causing temporary nicks. So there is loose. So it can be easily separated by helicase enzymes. Helicase enzymes simply cut open that strand. Then single-stranded binding protein, SSB protein, will coat each strand. So there is no chance of rewinding. That's the role of single-stranded binding protein. The other is primase. Primase simply synthesizes a primer, which is complementary to DNA strand. The reason is it provides 3-hydroxyl group to start what you call replication. After that, there will be adding of nucleotides. That is what you call elongation. Simply DNA polymerase III add nucleotides to the 3-hydroxyl end of the primers on both leading and lagging strand. Lagging strand. So primers are removed by exonuclease activity of DNA polymerase I. Then the gap will be filled again by DNA polymerase II. The question is, why must the lagging strand be produced in discontinuous in Okazaki fragment? Why it not synthesized continuously like leading strand. Because in the lagging strand, the end of growing strand faces away from the replication fork. Away from the replication fork. Away the replication fork. So that it is in the manner of discontinuous. It is in the manner of discontinuous. So fragment is formed. The gap will be formed. That gap is filled by ligase enzyme. The ligase enzyme. Ligase enzyme, it fills the gap, which help in formation of phosphodiester bond. Look at here. Here is topoisomerase enzyme. Look, this is a topoisomerase. This is a strand of DNA. So replication begin at the origin of replication. Then the Bible will be formed. After that, helicase separate the strand. This is helicase enzyme. Topoisomerase enzyme, it remove supercoiling, so that helicase enzyme easily cut the bonds. Then single-stranded binding protein bind each strand. So it never coiled again. It never twisted again. That is the role of single-strand binding protein. Topoisomerase removes supercoiling. It makes it loose by making temporary nicks. Here, DNA polymerase simply synthesize, add a nucleotides. There is what you call sliding clamp. This sliding clamp, it holds DNA polymerase 3 at this position while it's doing its job. DNA polymerase 3, it add new nucleotide, it synthesize new strand. So sliding clamp, it hold this DNA polymerase 3 at this position. In such a way, replication can be carried out. Look at here. This is 3 to 5 direction. This is 5 to 3 direction here. So upon this strand, leading strand synthesize continuously toward replication fork from 5 to 3 direction. If this is from the 5 to 3 direction, since the strands are antiparallel, this strand will be from the 3 to 5. So it will come and it synthesize like this. Then after it finish, it come again and synthesize. After it finish, it come and synthesize. After it finish, it come toward replication fork and synthesize. So the gap will be formed. This gap will be filled by DNA ligase enzyme. This is a DNA ligase enzyme. DNA polymerase 1 and DNA polymerase 2 is very important in repairing mechanism. This do have exonuclease activity. It remove primers, so it seal that area. Primers synthesize a primer. After primer is synthesized, it removed. Synthesize RNA primer begin the replication, then it will be removed. After it will be removed, DNA polymerase 3 do this activity. Mind you, this is in the form of Okazaki fragment, which is lagging strand away from the replication fork. That is a point. So how this DNA replication can be terminated? Replication needs to know how to stop. There is different signals. The cell know how to begin and how to stop. There is what you call tear sequence here. There is a tear sequence. Suppose this is an example I provided here. Tear sequence at the tear site, which is opposite to the origin of replication. approximately 24 base pair long. So there is also an enzyme that supports this tier sequence or tier site here. Tier site. So TUS is nothing, termination utilization sequences. It forms something complex. It forms something complex here. So this TUS aid in stopping what you call replication. Suppose this is a bubble is formed, replication begin, and it moves from all direction. This direction, here is tier A site, here is a tier C site here. TUS form complex here, again TUS form complex here. So these TUS involve in recognizing termination signal, utilize tier sequence to prevent replication movement. So the binding of TUS to tier site forms a protein DNA complex that arrests DNA replication. So in such a mechanisms, it stops DNA replication. It stops DNA replication actually. So with the help of TUS to tier complex, it stops what you call DNA replication. Thank you for watching this video. Subscribe, share, and comment. Thank you very much.